Welcome to The Explainer. Today, we're diving deep into Prescottella equi. Now, you probably know it better by its old name, Rhodococcus equi. This bug is a real problem. It causes a severe pyogranulomatous pneumonia in foals, and for immunocompromised humans, it's a huge threat. Just so you know, this entire briefing is based on a report put together by Gemini Deep Search. And if you want to check out the primary sources yourself, they're all in the linked notebook. So let's kick things off with what's probably the biggest headache for the clinical lab right now. What on earth do we call this thing? And trust me, this isn't just some minor name change. We're talking about a taxonomic journey that's been going on for 100 years. It started its life way back in 1923 as Corynebacterium equi. Then in 1977, it became the Rhodococcus equi we all know, and now, as of 2022, it's been officially rechristened Prescottella equi. So here's our game plan. We're going to walk through this just like a clinical microbiologist would. We'll start with the new name and how to report it, then we'll hit the bench for identification, talk about why molecular diagnostics have to be host-specific, get into the tricky business of treatment, tackle the AST breakpoint dilemma, and finally, wrap up with infection prevention and control.
All right, so let's get into the weeds on this name change. You know, this didn't just happen out of nowhere. It's all thanks to phylogenomics, which is basically using the entire genome to figure out who's related to whom. And when scientists did this, they took a really deep look and realized, hey, Rhodococcus equi doesn't quite fit in with the rest of the Rhodococcus family. It was kind of the genetic odd one out, enough to get its own branch on the family tree.
Okay, so a new name is scientifically correct. Great. But here's the real-world problem it causes, right? For more than 40 years, every single clinician, every vet, has known this bug as Rhodococcus equi. You can't just flip a switch overnight. So to avoid some serious confusion that could actually hurt patients, the best practice right now, the consensus, is to report it using both names. Something like Prescottella equi, formerly Rhodococcus equi. It just bridges that gap between the official taxonomy and what people actually know. Okay, enough of the names. Let's head over to the bench and see what this thing actually looks like in the lab. This is where the real fun, the real detective work, begins.
So, you get it under the scope. And what do you see? You've got these gram-positive coccobacilli. But the key thing here, and it's a big one, is its pleomorphism. It's a bit of a shapeshifter. In a young culture, it looks like rods, but in an older one, it looks more like cocci. And to make things even trickier, you can sometimes see it hiding out inside macrophages. All this variability, well, it sets a pretty big trap for the unwary.
And this right here is exactly where things can go wrong. That diphthroid morphology makes it a dead ringer for your everyday Corynebacterium species. You know, the stuff we often write off as contaminants from the skin. And that's the tragedy, isn't it? The report comes back saying commensal flora, but what's really happening is a life-threatening infection in an immunocompromised patient is being missed, and the clock is ticking.
Okay, so let's look at the culture plate. Here, the picture gets a little clearer. It's an obligate aerobe, so it needs oxygen, and it'll grow on your standard media. But it takes its sweet time. You're probably not going to see much for at least 48, maybe even 72 hours. When they do pop up, the colonies are usually kind of mucoid and non-hemolytic. But the real tell, the thing you're looking for, is that classic salmon-pink color. It starts faint and then gets this beautiful coral red hue as it sits.
Now, one of the classic biochemical tests you can use to nail this ID is the CAMP test. You take your unknown, streak it at a right angle to a beta-hemolytic Staphylococcus aureus, and if it's Prescottella equus, you get this awesome-looking shovel-shaped area of enhanced hemolysis. It's a really neat reaction, and it's all thanks to an enzyme called cholesterol oxidase, which comes from the CHO-E gene. Now remember that gene, CHO-E, because it's absolutely crucial for what we're about to discuss next. All right, let's switch gears to molecular diagnostics. And folks, this is one of those areas where you really have to pay attention, because the line between veterinary and human medicine is crystal clear. Grabbing the wrong molecular assay here isn't just a simple lab error, it's a surefire way to miss the diagnosis completely.
So here's the absolute bottom line. You cannot use the same test for animals and humans. It just doesn't work. See, in foals, the disease is caused by isolates that have this specific virulence plasmid. So vet labs rightly go after the VAP-A gene, which is on that plasmid. But here's the twist. In humans, something like 3/4 of the isolates don't even have that plasmid. So if you want a reliable ID from a human sample, you have to target a gene that's always there, on the chromosome, like that CHO-A gene we just mentioned. If you use a VATS VAP-A PCR on a human isolate, you're going to get a false negative. It's a massive, massive miss.
Okay, so you've identified it correctly.
Now what? How do you treat it? And this is where things get really complicated really fast. The stark reality is this. There are 0 formal treatment guidelines for humans. Nothing from the IDSA, nothing from ESCMID, so clinicians are basically flying blind, piecing together treatment plans from case reports and expert opinions instead of solid clinical trials.
Believe it or not, the only place you can find actual structured guidelines is in veterinary medicine. Take a look at these from the Dutch Formulary for Horses. You can see a clear pattern, right? It's always combination therapy, it's always for a long time, and they use drugs that get inside the cells really well. Think macrolides plus rifampicin.
Which of course leads to the million-dollar question. What do you do for our human patients? I mean, you can't just take the treatment for a horse and give it to a person.
So the strategy in humans is based on a few core principles. Number 1, always use combination therapy, at least 2 drugs that work. Number 2, you need drugs that can get inside the cells, where this bug likes to hide. And number 3, treatment is usually long. We're talking months, not weeks. 2 to 6 months is pretty standard. Your go-to agents are often things like vancomycin, carbapenems, linezolid, and rifampin. But there's a really interesting debate going on now about whether we really need such long courses, especially for patients whose immune systems recover.
Okay, get ready for another major hurdle for the lab, antimicrobial susceptibility testing, or AST. And I'm telling you, this is an area where the communication between the microbiology lab and the clinical team has to be absolutely rock solid.
The problem is actually pretty simple, but its impact is huge. As you can see here, EUCAST has not set a single clinical breakpoint for Prescottella equi. Not one. And if you don't have breakpoints, you cannot, under any circumstances, report S, I, or R. Full stop.
So what are you supposed to do? The official guidance from EUCAST is clear. You run your AST, you get the MIC value, that number, and that's what you report. Just the number. No interpretation, no S, I, or R. It's then up to the clinical team to take that raw MIC value and figure out what it means for their specific patient. And ideally, they're doing that while talking to you in the lab.
So how do they interpret it? Well, this is where a slide like this becomes your best friend. These are published MIC distributions. This data essentially becomes your reference point. You look at the MIC from your patient's isolate, compare it to these typical ranges, and you can see, is this a wild-type bug, or does this MIC look unusually high, suggesting it might have picked up some kind of resistance?
Okay, let's wrap this up by talking about infection prevention and control. Now, when you think about where this bug lives, you're probably picturing a dusty field, maybe a horse farm, right? Soil, manure, that kind of thing. But we need to look somewhere else, somewhere much closer to home, and honestly, somewhere a lot more concerning.
Yeah, that's right. While we're all thinking about farms, the CDC has pointed to a threat that's literally inside our hospitals. We're talking about the plumbing, the water pipes in the walls. And this is a perfect example of a modern IPC challenge. The CDC has actually given it a classification. It's an OPPP, an opportunistic pathogen of premise plumbing. That's a fancy way of saying it loves to live in our water systems. Sinks, drains, showerheads, you name it. It can set up shop and create this ongoing risk of hospital-acquired infections. And the takeaway here is unavoidable. Every single hospital needs a serious, robust water management program.
So, let's just pull this all together. We've seen how a simple name change is actually about deep genetic science. We've seen why diagnostics absolutely have to be different for a horse versus a human, and now we know that infection control isn't just about hand hygiene, it's about what's in the pipes. It all brings us to this final, really important question that we should all be asking ourselves back in our own labs and hospitals.
Hey, if you got something out of this briefing, do us a favor and subscribe to the channel. We're really trying to build a community of practice here. So jump into the comments. I'd love to hear how your lab handles Prescottella equi. What are your protocols? What challenges are you seeing? Seriously, sharing your experience helps the entire community get better.